mRNA caps are little biochemical structures that are at the very front end of an mRNA. An mRNA is a very linear molecule. It's got a front and a back. And the front end we call the cap. What that cap structure does is it protects the mRNA from degradation and also helps that mRNA get translated into a functional protein. So we recommend CleanCap over ARCA and enzymatic capping because CleanCap is going to allow you to get a Cap1 mRNA structure at the highest possible yield and capping efficiencies relative to ARCA or enzymatic capping. ARCA and some other co-transcriptional cap analogs that came before only could get you to the Cap0 structure and frequently suffered from low yield because you had to starve the reaction of key raw materials that are needed for mRNA manufacturing. Enzymatic capping is chosen by a lot of users, but has a disadvantage where it requires subsequent manufacturing steps. First, you have to make the mRNA by in vitro transcription, and then you have to go to a second biomanufacturing process to add that cap as a second bioreactor and a second enzymatic step. Those extra steps increase the overall labor and time that you're spent, at the bench or in a bioprocessing space, rather than being able to do all of that at once in a single co-transcriptional capping reaction. So the CleanCap technology is available in two formats. We manufacture the cap analog and have that available for scientists that want to manufacture their own mRNA, whether that be in a research use environment or ultimately wanting to scale that up into some sort of clinical space. We have RUO and GMP grades of CleanCap available. The alternative way to get access to CleanCap is we will manufacture the mRNA for you. We have a full-service CDMO that goes from research scale up to GMP phase 1, and we're able to do that manufacturing services for you to make mRNA with CleanCap. CleanCap has an advantage in vaccine development in that it's a co-transcriptional capping strategy that's going to get you a Cap1 structure in the highest possible yield. The reason that this is advantageous for vaccine development is it's going to allow you to scale your process to meet the demand of making hundreds of thousands, if not millions of doses of a vaccine to curtail various infectious diseases. CleanCap was used in the COVID-19 mRNA vaccine response and has ultimately helped those processes scale to make billions of doses of the COVID-19 vaccine that has been administered to date. We believe that CleanCap can confer this advantage to all vaccines that are currently developed using mRNA technology. The advantage of using CleanCap in cell and gene therapy development is that CleanCap is going to allow you to have a co-transcriptional mRNA manufacturing process that gets you a Cap1 structure and the highest possible yield. By removing excess processing steps, you're going to streamline the manufacturing process of your ex vivo cell therapy, getting the appropriate mRNA raw materials to modify those cells, or if you're looking at a gene therapy, allowing you to scale that process to get the appropriate amount of mRNA needed to make an effective dosing of your gene therapy product. Overall, this is going to reduce the total manufacturing time needed to make your cell therapies or make your gene therapies, making them more accessible to patient populations.